Hello, my name is Chris Bohl, and I am the Products Technical Support Manager at XenoTech. And today I want to talk a little bit about what are subcellular fractions and which ones should you use for your experiments. But before we get into this, we should first talk about what subcellular fractions are. There are various subtypes of subcellular fractions, but a general description is that they are non-living in vitro matrices that are comprised of the tissue's cellular components and enzymatic activities. The different types of subcellular fractions have undergone various centrifugation steps to either enrich for or diminish specific activities found within the source tissue. The vast majority of the most commonly used subcellular fractions are comprised of fractions that are pooled from many different donors. This is done to minimize donor-dependent phenotypes, as individual donors or small pools can skew data to only be representative of the donors in the pool and not necessarily the activity that you might find in a larger target population. But that is not to say that the individual donors or small donor pools aren't useful, because they are. They just have more specialized uses compared to the larger pools. So let's talk about how the different subtypes of subcellular fractions are made and what cellular important components and activities each fraction contain. This simple diagram starts with liver tissue as our source of the cells. However, most subcellular fractions are made using these general steps, and the starting tissue can be exchanged pretty freely. The first step in subcellular production is to break up the tissue and homogenize or lyse cells in a buffer to release the various cellular components. Once the tissue and the cells are homogenized, it is centrifuged at low speed to clarify the homogenates and get rid of the unbroken cells, connective tissue, fats, and intact nuclei. This fraction is what we call the tissue homogenate. This fraction is the most complete as it contains the vast majority of the tissue components and enzyme activities, with the exception of the components that were removed during clarification. The homogenate is then centrifuged at medium speed to create the S9 fraction. The S9 fraction, the supernatant, is collected and contains enzymatic activities from small vesicles, microsomes, and soluble cytosolic proteins. The pellet, which is most of the time disposed of, contains larger intact organelles like endosomes, lysosomes, mitochondria, and large membrane fragments or vesicles. The S9 fraction can then be centrifuged at high speed to separate the microsomes from the soluble cytosolic proteins. The supernatant is the cytosolic fraction, and the pellet contains the microsomes. After the cytosol fraction is pulled from the microsome pellet is washed and repelleted to further enrich the specific activities associated with the microsomal fraction. So who uses these fractions? It is generally used by scientists that are doing preclinical drug development work. A majority of the work of these reagents are used in a pre-IND filing. And what are they used for? I just mentioned that the vast majority of the subcellular fractions are used for in vitro ADME, DMPK work, like examining compound stability, identifying the major enzymes involved with a compound's metabolism or reaction phenotyping, metabolite characterization, CYP inhibition, or making comparisons between species so you can pick the most relevant small animal model. They're also useful for potential gender differences in all the above type assays, contributions of genetic polymorphisms, toxicity predictions, DDI predictions, and IVIVE modeling. While it is said that hepatocytes are the gold standard for in vitro work, and they are, there are some pretty significant advantages to using subcellular fractions in your research. They are easy to use, they're significantly cheaper, and have individual lot volumes that single donors or even pooled hepatocytes can't come close to reaching. This gives researchers long-term lot availability and reproducibility. You can get matching S9, microsomes, and cytosol from clinically relevant first-pass tissues and from all the relevant laboratory species. Our production protocols have been vetted and our products have been used by the industry for decades. We are also in an exceptional position where our products also have a second layer of quality control, as our services CRO uses the exact same products in their sponsored studies that are available for researchers in our field to purchase. There are numerous options that are available, such as pools of donors that have specific disease states, genders, specific genotypes, donors with high or low specific activities, pools with high AO-XO activity, large pools, or really large pools. Our two most popular human liver microsome pools fall within the large or really large pools category and offer different advantages. The most popular pool is our Extreme 200 pool, comprised of 200 donors that are equally represented in both gender and individual donor tissue contributions. The donors in this pool are randomly chosen and represent the general U.S. population, as any donor in this pool can be swapped with any other donor in our inventory without significantly affecting the average activities. The Extreme 200 pool is produced in large lot volumes, offering sustained long-term availability. The pool of 50 is a little bit different, as donors that make up this pool of 50 are pre-screened for enzymatic activities and are not equally represented. It is pooled in a way to hit specific average activity windows, and this pool is made in smaller volumes, although still significant amounts are made. And it comes with slightly less characterization than our pool of 200. As mentioned previously, subcellular fractions from small animal models can be extremely useful when doing screening and species comparisons to make sure you choose the most relevant small animal model for in vivo and toxicology studies. I also want to reiterate that while the liver is many times the primary tissue responsible for xenobiotic metabolism, there are other tissues and tissues that are significantly contribute to xenobiotic metabolism and should be taken into consideration when choosing which subcell to use. What is the physical characteristics of your compound? What tissues and pathways is it trying to target? And how is it going to be introduced to the body? Is it going to be ingested, inhaled, applied topically? Is it going to be injected intravenously? These are the few things that might steer you towards choosing an extrahepatic subcellular fraction for your work. Like hepatic subcellular fractions, extrahepatic subcellular fractions are available from different species, and at least with humans, the lung subcellular fractions are split between pools that are produced from donors who smoked and those that did not, as it's been long known that smoking can affect the expression of certain P450 enzymes in the lungs and may have a significant effect on metabolism. Because I mentioned that there are differences in our human lung subcellular fractions, I also want to take a moment to explain the two options available with our intestinal subcellular fractions, as the difference between the two can have a significant impact on your data and its interpretation. Intestinal subcellular fractions production is unique in that it requires the use of protease inhibitors during tissue processing. This is due to the detrimental effects of the high levels of endogenous proteases present in the lumen and mucus of the intestine. Without the protease inhibitors, the proteases would catabolize the intercellular enzymes as the enterocytes are homogenized and lead to subcellular fractions that contain decreased metabolic activities. The downside is that the PMSF in the inhibitor cocktail can and will inhibit the endogenous esterase activities to some degree. If the PMSF is left out during production, the metabolic capacity of the subcell is decreased, but the esterase activity is left uninhibited. So, if you predict that your compound will be mostly metabolized by cytochrome P450s, you'll want to use the standard PMSF-containing intestinal subcell. But if you think that your compound will be metabolized by intestinal esterases, the PMSF-free subcell will be the better choice for your experiments. I also want to point out that the intestinal subcell is the only tissue where protease inhibitors are used during production. No other subcell fraction from any other tissue are produced in the presence of protease inhibitors, so altered esterase activities in other subcell fractions are not a concern. So the subcell fractions that we just talked about are the classical standard subcellular fractions, and they are useful for monitoring metabolism of many types of compounds. However, they aren't always as useful for looking at the metabolism of larger molecules or biologics. Compounds for which endocytosis and trafficking through the lysosome is a major route of metabolism and/or catabolism. For studying these types of molecules, we have purified rat liver tritosomes and human liver lysosomes to monitor lysosomal stability and metabolite formation in an in vitro experimental design. Tritosomes and lysosomes are used for many of the same kinds of assays traditional subcells are used for: stability, metabolite characterization, and prediction of safety, but just with different classes of molecules such as ADCs, oligos, recombinant proteins or peptides, nanoparticles, and antibiotics. Rat liver tritosomes are modified lysosomes that have been loaded in vivo with triton. This modifies the lysosome's density, making them much easier and efficient to purify them through sucrose. Tritosomes have high specific activities for lysosomal specific catabolic enzymes. Human liver lysosomes are isolated in their native form and also have high specific activities for lysosomal enzymes. They are characterized for the same activities as rat tritosomes are, but are more representative of the lysosomal compartment than alternatives such as acidified S9. Okay, so now we have a good idea of what, how, who, and why of subcellular fractions. We should take a moment to go over which subcell fraction might be the best for your experiments. Homogenates offer the most complete tissue proteome. However, it has the lowest specific activity of enzymes that are mostly of interest for the ADME DMPK field. S9 is a combination of the microsomes and cytosol fractions and contains the majority of the phase 1 and phase 2 activities, and it has higher specific activities than homogenates. With S9, you can spike the incubation with a mixture of cofactors, UDPGA, PAPS, GSH, along with NADPH, and you can examine multiple cofactor-dependent metabolic activities in the same incubation. Microsomes are useful if you're focusing on phase 1 and UGT enzymes, as they are highly enriched in these fractions and have the highest specific activity of the common enzymes of interest. Cytosol contains mostly phase 2 enzymes and soluble phase 1 enzymes, but are depleted of the activities associated with the microsomes. And don't forget, if you're using intestinal subcell, standard PMSF containing subcell for all non-esterase activities and PMSF-free subcell for esterase activities. Lysosomes and tritosomes are for compounds or molecules that in vivo are expected to be endocytosed from the cell surface and trafficked to the lysosomes. We also get questions from customers about whether to use an NADPH regenerating system or just spike the incubation with a final concentration of NADPH. At XenoTech, we like to use a regenerating system in all of our incubations that require NADPH as a cofactor. Regenerating systems will maintain a steady level of NADPH for up to 4 hours at 37 degrees. So no matter how long you decide to take your incubations out to, you have one less variable to have to think about. We have been making and using these fractions in our own sponsored services for over 25 years. And we've been doing it so long that our phone number is actually GET P450. For those of you who still remember the catchy alphanumeric phrases that used to be used to try to get people to remember your phone number, we still have it. But just in case you can't find what you need commercially, custom products and custom preparations might be an option to explore. We have a designated department just for custom preparations, and they've made some pretty unusual products over the years. Something as simple as custom volumes of standard products, all the way up to insect microsomes and some interesting cellular isolations. It's also useful to look through our custom product surplus inventory, as there might be something left over from a previous custom preparation that might fit your needs without having to wait for a new preparation to be fulfilled. Last, but certainly not least, I want to assure everyone that all of our tissues are sourced from accredited and licensed sources, along with a full consent for research in the instance of human tissue. We do not accept tissues that could be used to save lives, and they are negative for any major animal or human pathogens, including SARS coronavirus 2. With that said, we still recommend the appropriate PPE because when working with these reagents, we consider them BSL-1. However, caution should be taken. We also have official statements prepared that outline how we obtain these tissues, and we are more than happy to help share these documents with researchers to show their regulatory groups and/or to send to journals to satisfy ethical requirements for publication. Also, all of our primate subcellular fractions come with CITES permits for those who are outside of the United States. These are just permits that are required that show tracking of the monkey tissue. So thank you for your time, and if you have any questions or would like personal recommendations for products, you can email me or one of my team members, and we'll do what we can to help move your research forward. And thank you for your time.